Hello and welcome back to all of you who joined us yesterday for day one of the course. Um, if you weren't here yesterday, I'm Sean Gibbons, a faculty member and microbiome researcher here at the Institute for Systems Biology in Seattle, or ISB for short. ISB is a nonprofit biomedical research institute. We are an affiliate of the Providence healthcare system. We focus on some of the most pressing issues in human health. Systems biology enables us to better understand the human microbiome, like we'll hear about today, but also cancer, aging, brain health, chronic disease, infectious disease, and more. ISB also focuses on K through 12 STEM education. And today we'll be looking at beyond K through 12 education, right, for a lot of grad students and postdocs and so on. We are interested in particular in addressing issues of inclusion and equity for all students, including historically underrepresented groups in science. You can learn more about ISB by visiting our website, isbscience.org. As a nonprofit research organization, we rely on the generous support of our donors. So thanks to those donors. We put on a number of virtual events every year, and we have two coming up in November that I'd like to point your attention to in case you're interested in joining us. On the evening of November 13th, we're hosting an in-person event called Ignite STEM, and it celebrates the impact of our ISB education team on teachers and students around the US. On November 21st, at noon Pacific time, we will host our final research roundtable of the year. ISB Associate Professor Dr. Wei Wei will talk about his new technique that his lab has developed to provide real-time information to physicians to inform precision cancer treatment. So we put on these events at no cost to registrants. If you've got some change rolling around, you want to throw our way to help support this kind of work, feel free to donate. There is a donate link at the bottom of the course website. We hope everybody had a great Indigenous Peoples Day back on Monday on the 14th here in the US. I'd like to just recognize that we're on the traditional land of the Coastal Salish, in particular the Duwamish people, and we honor with gratitude the land and the people, past and present. All right. So then the next thing to get into is our generous sponsors. We have 3 sponsors this year of our event. These sponsors allow us to provide this event to everyone free of charge. First up, Applied Microbiology International not only provides some financial support to this event, but also they are providing free memberships to everyone who registers for the Microbiome Series this year. So please take advantage of that. Environmental Health and Microbiome Research Center, or the Embrace Center at the University of Washington, is another one of our sponsors. And then finally, we're sponsored by Science in Seattle. Like I said, because of their support, we've been able to provide this to everyone for free this year, but also for the past 4 years. So this is our 5th year of running this microbiome series. And overall, we've had 6,000 registrants from about 80 countries. So thanks to everybody for your engagement over the years. To all of you tuning in from around the world, we're really grateful for your time today. We're excited for this opportunity to train the next generation of microbiome researchers. I feel like as an elder millennial, I'm perhaps out of touch with what is currently used in terms of social media. We used to have hashtags and stuff. So if you're still on X or Twitter, we do have a hashtag for sharing your experiences today, #ISBMicro24. We probably should have had a TikTok channel for this, but I'm not savvy enough for that. Maybe in the future. We're going to launch a poll here shortly to assess everybody's career stage, kind of seeing where you're all coming from. So stay tuned for those results. In the meantime, I'll give a little bit of a primer on what you can expect today. So today is the 2nd day of the course, and over the past several years, we've kind of dedicated the first day to giving people the basic tools to process and analyze 16S amplicon sequencing data from raw sequencing data up through statistical analysis. And that was Alyssa Easton yesterday, your instructor, giving you that course. Today is going to be on metabolic modeling, right? So once you have assessed the community composition of a sample, You want to know something about what's the functional capacity of the system, like what's coming out of that microbiome in a given context, like on a given diet, for example. So Dr. Alex Carr will be walking us through today as our instructor, a course on metabolic modeling or community scale metabolic modeling. I want to give an intro to our whole team. So in addition to Alyssa Easton and Alex Carr, who've been our instructors over the past couple of days, we have a lot of folks behind the scenes helping out. So as teaching assistants today, we have Jacob Cabin, Noah Rappaport, Alyssa Easton, Crystal Perez, Carl Geiser, and Nick Quinn Bowman, all helping out on the Slack. Our admin support, our coordinator is Dominic Lewis, Allison Kudla, Joe Meister, Audrey Hubbard, Erica Krager, Ray Rosario, and Thea Swanson. And without these people, none of this could happen. All right. So now I think we'll quickly go back to the poll to check out where everybody's coming from. I'll wait for that to pop up on my screen. All right. So looks like, as expected, the majority of our registrants here today are graduate students. 47% of everybody joining us is a graduate student, 23% are postdocs. We have 7% industry scientists, 12% educators, 2% undergraduates, you intrepid undergrads, thanks for joining. And then other is 9%. So I know in the past we've had Instagram influencers joining us on this course. So yeah, if you're out there, maybe make us go viral. That'd be great. Okay, and so then as I approach the end of my prepared comments, I'll just say that we've been doing this for 5 years. It's been a lot of fun. We've got a lot of great feedback, and I think it's been an important service to the community, especially back during the pandemic when we couldn't get together and have these kinds of classes in person. Having these virtual tools allowed us to kind of disseminate this knowledge more widely. But I think now, after 5 years, there's a lot more options out there for these types of virtual symposia, virtual courses. So I think we're going to wind down our end of this. This will probably be our last year of teaching the course. We might reboot it someday in the future. We'll probably keep having our symposium, but you're fortunate to be with us this year because it is, we're ending off on a high note with this theme of the gut-brain axis. So let's get into the course. If you have issues today, you can visit the Get Help channel on the Slack, and our TAs will be on top of getting you help. You've already received a link for the Slack in the email you got when you registered, so you can go click on that to join the Slack if you haven't done that already. To ensure everyone can enjoy the course in a safe and harassment-free environment, we provided a code of conduct, which can be found pinned in the general channel on the Slack. If we observe behaviors that violate this code, we will warn perpetrators, and we reserve the right to remove folks if the behavior persists. Again, this has never happened in the past. Everybody's very well behaved. Just letting you know that this protection is in place. All right, and so without further ado, I think we'll just move along. Alex Carr will be your instructor today. You're in wonderful hands. It should be a really fun day. And then don't forget that on Friday, we're having our symposium where we're going to see scientists from across the world who work on the gut-brain axis field. And we'll see some of the kind of cutting-edge research in this area on Friday. We will start the course at 9:30 a.m. Pacific time sharp. So as soon as I end off here, we'll kind of have a break. You can go, use the bathroom, get a drink, and then we'll see you back here at 9:30 a.m. Pacific for Alex's start of the course. All right, see you soon.
All right, welcome back. Thank you for joining us for the second day of this year's ISB Microbiome course. My name is Alex. I'm a postdoc in the Gibbons lab, and I'll be the instructor for today's portion of the course. The focus of today's course is predicting microbiome activity in the context of Parkinson's disease with metabolic models. So let's get right into it. Before we get started, let's make sure you're set up with the course notes. You can open these slides using the URL shown here, which should also be in the email that was sent to you prior to the course. The TAs will also be posting this link in the Slack channel, so you can find it there as well. As a reminder, as with yesterday, this course will be taught across 3 modules. The presentation slides we're currently looking at will contain explanations of the logic and methods used in the course, as well as links to pertinent materials related to the content included in the course. The analysis and modeling, as well as visualization of the results, will take place in the course notebook, hosted in Google Colab. You can navigate to the notebook using the link in the next slide. Let's see, also, there we go. Or from the email sent to you before the course. Finally, the Slack chat is available to you at any time during the course for questions or comments. So if you have any questions, don't hesitate to post them there and the TAs will be happy to help you out. For this next part of the course, we'll revisit the theme of this year, of the course this year, microbes and their impacts on our minds. So how do microbes influence our minds? And how can this impact the disease progression in the case of Parkinson's disease? Well, as Alyssa pointed out yesterday, the microbiome is a source of a wide range of molecules that can directly or indirectly impact host processes. One class of microbiome-mediated metabolites, short-chain fatty acids, or SCFAs, have garnered a lot of attention in recent years for their apparent anti-inflammatory properties. As this figure demonstrates, a variety of factors, including diet, age, environmental exposures, and immune status can impact the state of the microbiome and the production of SCFAs. Diets high in fiber and other beneficial metabolites like polyphenols, omega-3 fatty acids generally lead to higher levels of SCFAs in the gut, which promote gut barrier health, lower gut inflammation, and can have downstream effects that ultimately impact brain function. Additionally, fiber promotes gut motility, which is important as constipation is one of the hallmark symptoms that precedes the development of Parkinson's disease. Acetic acid, propionic acid, and butyric acid are the three most common SCFAs produced, representing around 60, 25, and 15% respectively in humans. Estimates suggest they are responsible for between 60 and 70% of colonic epithelial energy in humans. And in addition to their anti-inflammatory properties and uses as energy sources, butyrate and propionate specifically have been shown to impact the production of hormones that can suppress feelings of hunger through signaling interactions that are mediated by the FFAR3 receptor on the nodose ganglion, among other beneficial properties. Thus, SCFAs play a role in a wide range of processes that can benefit the host. Before we jump into the material, let's follow the link to the notebook and familiarize ourselves there. So I've already pulled it up, but you can also click this link. And here we are. Before we get started with the notebook, you're going to want to go here to the file and save a copy to your drive. And then you can see it opens a new tab. I'm going to go ahead and close this one. And then I'm going to call this one Alex's. Great. And then you don't have to, but I'm going to click here to connect to Google Colab. If you run any of the cells, it will also achieve the same results. So as before, yeah, the notebook is divided into different sections. You can use the table of contents to see what we're going to be covering. You can see, for instance, there's the setup, the use of this tool, MyCom, which we'll explain in more detail, some visualizations, and then some exercises at the end. You can also use this little folder icon to see what files are available, which currently there's none. So we need to get started by cloning the GitHub repository and then moving into this materials folder that's being downloaded. So as I open this, now you can see there's this materials folder that is being propagated with all our delicious goodies from the GitHub. And it looks like we got everything. Next, we're going to want to install MyCom and enable Chime 2 interactions, as we will be using the tables that were generated in yesterday's portion of the course to produce the metabolic models that we will be using to simulate gut microbiome activity. If you weren't here for the yesterday's course, don't worry about it. We're actually going to just be pulling these files directly from the treasure chest. So they have already been produced for everybody. And that's partly because the Google Colab actually will disconnect and erase all the files after a certain amount of time. So we wouldn't expect you to still have those available. Great, it looks like everything installed. This figure gives you a nice overview of some of the functionality of MyCom, and it might seem a bit complicated, but don't worry, I'll explain it in detail here in a moment once we get back into the slides. But before we do that, we're gonna start the process of getting the model simulations going so that while I discuss or cover the theory behind it, we'll have plenty of time to ensure that the results are ready for us when we come back. So just to get started, we can import some test data and look at it just to see how a typical table might look that is going to be used or fed into our simulation tool. Here, for example, you see we have two different samples, sample 1 and sample 2. Sample 1 has 4 identical strains of E. coli, and you can see it lists their names. Here, they just have the arbitrary species names, E. coli 0 through 3. It also tells you some information about the models, how many reactions are contained, how many metabolites are part of those reactions, and their abundance, which is very important as it'll determine their individual contributions to the overall community activity. Great. So now I think we are ready to import our data from yesterday. So you can see here we're using this function chime to MyCom, which is taking the QZA tables that were generated yesterday, one of which provides the abundance of the different features that were identified and the taxonomy. And for the purposes of today, we'll be focusing on the genus level taxonomy. Generally, with 16S sequencing, genus is the best resolution that we can get. And by that, I mean, we can get a decent quantity of the data mapping to the genus level. We would expect maybe between 60 to 80 or more percent of the total relative abundance mapping to genus. We'd like to work with species, but unfortunately, 16S sequencing doesn't allow us to reliably detect species. So here we have our genus table. You can see again, sample IDs. We have the abundances, the genus name, which in this case matches the ID, and the relative abundance. Next, we're going to import our metadata and merge that with our taxonomy table using pandas, which is a great tool for working with tables like this, and one you're probably already familiar with from yesterday's portion of the course. So now you see we have all of the metadata. Now we're going to get into the meat of our metabolic modeling. Here, we're going to run the cell to start building our models. This process takes our taxonomy table as well as this Agora genus QZA file, which provides information about the models. We're using models from the Agora 1 database. which have been allocated at the genus level. So we're merging all the strain level models into a pan model at the pan genus model. And then it also tells what solver to use. This is very important as these are very complex problems that to solve. And ideally, we would want a very powerful solver to be able to get answers for these problems. Today, we're using this freely available one, OSQP. It's great for nonlinear problems. It's not amazing for the task that we're performing now, which is a very large linear problem. So one thing to make note is that because of that, the results that you get and the results that I'm presenting can be a little bit different, and I would expect that they will be a little bit different. Now, so this is running. It's going to take a little while. Go ahead and run these next couple cells as well, so that once this one finishes, it will run this one, which is importing the medium that we're going to be using. This is important because we need to specify to our models a representation of the diet. Here you can see we're using a representation of the Western diet. And we're doing this for simplicity because we don't have very much dietary information for these samples. But ideally, you'd want to have a customized diet for every sample if you knew what the diet was. Additionally, this step here is actually once you build your models and you have the diet, you can take those models and your diet to then perform the growth simulation, which is you can see is here. And then we'll have those results to come back to. Great. So like I said, this is going to take some time, between probably 15 and 30 minutes. So while that's running, let's jump back to the slides, and then I'll go over the theory behind this so that we can try to understand what we're doing. Great. So yesterday, we used QIIME2 to analyze sequencing data, starting from raw sequences, and ultimately generated the table of amplicon sequence variants, or ASVs, that we just imported and used to build and simulate, or start building and simulating our models. We also assigned taxonomy to our ASVs and used diversity metrics to compare samples. Today, we'll look at functional analysis of the gut microbiome. That is, rather than looking at what populates the microbiome, so looking at the genera, for example, we'll be determining what is happening in the microbiome, which is not always fully explained by taxonomy alone. When we first say functional analysis, a few things might jump into mind, like some of the omics methods listed here. Most commonly, to determine the function of the microbiome, one will do a metagenomic study from which you can see what genes are present in the community. You can also use 16S sequencing, which is what we presented on yesterday and what we're using today. But if you do have metagenomics data, then mapping these genes to a reference database is a simple task from which you can infer what reactions might take place in the system and put together a network of reactions, which might tell you about the functionality of the community. There is a caveat, though, in that gene abundance data doesn't always tell the full story. That's because gene abundance really only reports the metabolic capacity of a community. If you imagine a microbial community with a really high abundance of starch degradation genes, you might infer that a lot of starch degradation pathways are active. But if the host isn't eating any starch, then clearly this inference is wrong. Gene abundance tells you what could happen, but not necessarily what is happening. Additionally, when we're working with 16S data, we're making a bit of an inference step because we're needing to map our genus level inference of the taxonomy to a model at the genus level. But we're assuming that mapping is capturing like the reactions that are present in the cells that are in the microbiome, but this is a little bit of an inference, and we don't necessarily know that to be totally true, which is why if you can do a metagenomic study, that might be helpful in this case. All right, so gene abundances and other omics approaches are cool. For instance, I didn't talk about some of the other ones, but you could imagine doing like a proteomics study where you look at the protein abundances, or transcriptomics where you might look at the actual abundances of the transcripts to get a better idea of the functional capacity of the microbiome. But all these tools, they don't necessarily tell the full story. So today we'll explore an alternative approach to analyzing the function of the microbiome, predicting metabolic flux. Fluxes will tell us more about the actual metabolic behavior of the microbiome than abundances can. Great. So what are fluxes? Rather than abundances, which are a measure of concentration, fluxes are a measure of mass conversion into the system and through internal reactions. Since they represent a rate rather than a concentration, rather than an abundance, fluxes are measured in units of concentration per unit time. For instance, millimole per hour might be your unit of choice. In bacterial communities, we typically scale flux by relative abundance to account for differential metabolic contributions by more or less abundant taxa. So why am I claiming that fluxes are more informative than abundances? Well, if you look at the example on the right-hand side, which shows glucose going into glycolysis in the TCA cycle, we see that the cell in question is importing a ton of glucose, but that glucose is almost immediately being converted to pyruvate. If we were to measure internal glucose levels, they might actually be really low. This could lead us to incorrectly assume that glucose isn't required for the cell to grow. Obviously, this doesn't tell the whole story, since glucose is clearly being utilized. What would be more informative would be to see the flux of glucose into the cell and the resulting pathways that are activated and metabolites produced. Clearly, we can learn a lot from looking at fluxes in a system like this. The problem with measuring fluxes, however, is that it is a laborious task and can be quite costly. Longitudinal metabolomics can work, in which we measure metabolic abundances over time. However, again, this takes quite a bit of effort and can be expensive and is limited to those metabolites that are included in the metabolic panel. It also often requires growth in a defined medium, which in itself can be a huge hurdle. Isotopic labeling is another option using C13 or N15 isotopic labels, where we feed the community these labeled substrates using the natural isotopes that I just mentioned of carbon and nitrogen, and look for the appearance of those isotopes in other cellular metabolites, for instance, amino acids or other essential carbon metabolites. However, that can be even more costly and still limited to just those metabolites being investigated. So what we need is a way to easily and accurately estimate fluxes without the labor and expense associated with the methods that I just described. Luckily, there is such an approach. To do that, we can use a method called flux balance analysis, which has become more popular in microbiome research in recent years. This course is a testament to that. This is a computational method that uses genomic information to infer the most likely set of fluxes in a biological system. We can use this to predict the metabolic behavior of a microbe using genome-scale metabolic modeling. Let's take a look at how FBA works and how we can use that to infer these fluxes that we're interested in. FBA is a powerful computational tool that allows us to infer fluxes through reactions in a system by reducing the possible solution space of these fluxes to a biologically relevant one. FBA makes the critical assumption that the system being modeled, in this case, the metabolism of a microbe, is in a state of constant flow or steady state. This just means that the inflow and outflow of reactions in the system are equal. A good analogy to this is opening a window in your house. You'll have air flowing in and air flowing out, but you won't suddenly have more flowing in than out. If you did, then your house might expand and pop like a balloon due to the buildup of pressure, or condense and crumble due to the creation of a vacuum. That obviously doesn't happen. Thus, the steady state assumption mimics this and is applicable in several biological phenomena. If we look at our example reaction here, we have hydrogen and oxygen combining to form water. In steady state, we can say that stoichiometrically, the amount of hydrogen and oxygen consumed are equal to the amount of water produced. We can represent this mathematically with the first equation: reactants with negative coefficients and products with positive coefficients, balanced to 0. Repeating this for all the reactions in our system and factoring out the fluxes, we end up with a stoichiometric matrix designated S, as shown on the right. That represents all the reactions, as well as the unknown vector of fluxes designated V, which we are solving for. To satisfy our assumption of constant flow, we set the dot product of S and V, or S dot V, equal to 0, which sets up our system of equations. Additionally, we can further constrain the problem given known limits for the fluxes. For example, if we know the reactions are all irreversible, we might say that the bounds for the individual fluxes, the VIs and Js, are somewhere between 0 and maybe 1,000 units. Solving for a set of equations leaves us with a flux cone, as depicted in the figure on the right. The space in the multidimensional flux space that includes all sets of fluxes satisfying our constraints. You might notice, however, that there are an infinite number of solutions in our flux cone that could satisfy the problem that we've posed. So what's next? Well, the goal of FBA is to reduce that flux space to one that is biologically relevant, so that it mimics the actual behavior of the system as well as possible. But how do we find an optimal solution within the possible solution space? To find that solution, we can look at growth rates. We know that bacteria can only be present in the system if they can grow. Otherwise, they would be excreted and no longer exist in the system. In practice, this means we can say we want the solution in the flux cone that corresponds with maximum biomass, since this is generally the biological goal of a growing microbe. Biomass production can also be represented as a flux, since it is in essence a measure of mass conversion. What's great about flux balance analysis is that we can add information from a number of sources to constrain the problem and determine our optimal solution that satisfies this biologically intuitive solution. As we've mentioned, we can use the reactions present in the system to build a stoichiometric matrix, mathematically representing the reactions taking place and how they interact with each other. We can actually infer this whole stoichiometric matrix from the genome of a microbe by looking at gene abundances to determine what reactions are potentially taking place in the system, or in our case, by mapping our 16S genus identity to the closest matching genus pan-genus metabolic model. In practice, this means that the bacteria, so also, as we've mentioned, we make the assumption that the system is in steady state. In practice, this means that the bacteria is in the exponential growth phase, as noted in this growth curve. So that means that the growth rate is constant and biomass production is constant. So we include this assumption to arrive at a possible solution. Additionally, reversibility of reactions can be determined from thermodynamics, as many reactions can only occur in one direction, thus limiting the space of possible fluxes. Finally, we can add constraints to the import of metabolites going into a system, which can be leveraged to represent the metabolites' availability in the system to grow. This is a great way to represent the medium, as we just seen in the notebook. In practice, we can use this to model effects of different diets or different environments on the microbiome, adding relevance to the predictions being made by FBA. Cumulatively, these constraints help us find the region in the flux code that represents maximum biomass, as noted by this sort of red stripe in our flux code. The art of FBA is then to find a single solution in that solution space that is most biologically relevant. As you can see, that while there is still a reduced solution space due to these constraints that we've imposed, that solution space itself can still have an infinite number of solutions that meet our criteria. There are several methods available to find a single solution, but today we'll focus on these last two in the list, as these are the ones that are leveraged by the tool we're using, MyCom. One such method is minimizing the imports into a system. Since the microbiome can be a highly competitive environment, this is biologically intuitive, since the most successful residents in the microbiome would not need to rely on importing lots of metabolites to survive, rather subsiding on a smaller subset. Another common approach is parsimonious FBA, or PFBA, which basically minimizes the enzymes required to satisfy the constraints. Again, this is a biologically intuitive solution, since bacteria are efficient beings and don't like to expend more energy than is necessary on making enzymes. Really, though, the method you use is related to the question being asked and the system being modeled. Is it competitive? Is it resource limiting, etc.? I encourage you to look more into these methods if you're interested in conducting FBA on your own. Great. So now we've considered classic flux balance analysis used for genome scale metabolic models. And it seems pretty straightforward, maybe. Maximize the growth rate and get your answer. So you can just do the same in community models, right? Well, not quite. In genome scale metabolic models, we have a single growth rate. But in community models, we have multiple growth rates, one for each taxon, as well as a community growth rate calculated by multiplying the relative abundance of each taxon by its growth rate and summing them all together. If we try to maximize the community growth rate, you'll find we quickly run into some trouble. Classical FBA works fairly well when predicting the growth rates of individual bacteria. Here we can see how you can take a single organism and then either using 16S to map to a database of models or its gene content from a metagenomic study, you can infer the set of reactions that can then be used to represent the fluxes in the system, as shown here. And then you can use the approach that I just described of trying to maximize biomass under biologically relevant constraints to arrive at a solution that is both biologically meaningful and parsimonious. Additionally, what we can see is that generally, like for a single organism models, these can behave quite well, and the results of the simulation can, in some cases, agree very well with experiment. Here we can see on the right the results of a study that compared empirically measured fluxes with those measured by FBA across a wide series of evolved E. coli strains. We see that the predictions agree well with the empirical estimates, and that the evolved strains are able to achieve growth rates that are in the range of 90 to 95% of the theoretical maximum. In these two plots, the x-axis is the flux being predicted, and the y-axis is the flux being observed. And then the different colors represent different strains that have been evolved using optimization on these different pathways associated with central carbon metabolism. And on the right, you can kind of see like the optimality starting from the ancestor strain in black and then all the evolved strains and how those compare with the predictions, which in this case are quite good. The line here is the expected one-to-one relationship, and you can see most of these are pretty close to being on that line. So for single organisms, we can do a pretty good job. It's when we consider communities that things start to break down. Classical FBA doesn't do so great here. One of the issues is that given multiple taxa, optimal community biomass may be achieved by various linear combinations of the growth rate of the individuals. That is, there's a non-unique solution space for the growth rates and fluxes. So you might have a solution where the optimal community biomass is achieved by only one organism growing. Again, here we can see an example of how our community level model might be constructed, where you are now taking the reactions and forming individual models for the different strains in your community, and then putting them into a compartment that can represent the community model. And then again, doing the same, trying to do the same solution scheme. But unlike in the previous slide, where this was just a single line because you have a single organism, now this becomes this solution to the growth rate problem becomes an n-dimensional space where n is the number of strains. So if we have 3 strains, you can see now the solution space is growing quite large, and it will continue to grow exponentially larger as you include more and more taxa in your model. And you can see that leads to not only an inflated growth rate solution space, but also a greatly inflated flux solution space. Additionally, experimental results suggest optimizing community biomass, which is generally the objective that is used, may be a slightly flawed approach. As empirically, many communities achieve lower overall biomass as diversity increases due to competitive interactions, as shown on the right. Here we have 2 different kinds of communities, one composed of yeast, one of bacteria. And what we see is that as you increase the number of species from 1 up to 6, especially, this is true for both, but especially true for the bacterial community, you can see that the bacterial cell counts decrease with increasing a greater number of species, with the 6-member community achieving only about 60% of the biomass of the single organism community. So we do have a significant challenge here. But there is a solution, and this is where cooperative trade-off flux balance analysis comes in. This approach is able to generate unique, biologically relevant solutions to the community growth rate problem posed just a moment ago. Let's try to understand how. First, let's consider the growth curves on the left. As many of us are familiar, bacteria and other organisms typically start at a very low biomass and low growth rate, and increase their biomass until they have consumed all their resources. In doing so, they typically achieve a maximum growth rate somewhere along the way. As shown in the gray region, that eventually returns to 0 or close to 0. This growth rate trajectory can be mapped in a growth cone as shown below. Dynamic models typically are able to capture this trajectory, while classical FBA can provide a solution somewhere in the optimal region of the growth cone, but would not be able to contain it necessarily. CT-FBA, cooperative trade-off flux balance analysis, introduce a biological meaningful constraint by minimizing the distance between the point of 0 growth and the theoretical maximum, as shown on the right. This approximates the trajectory expected empirically. This constraint is achieved using an L2 style regularization that minimizes the sum of the squared growth rates of the community. This also empirically has the effect of distributing the growth rates more evenly across the community while allowing them to be unequal, whereas in a classical FBA approach, you will often get a solution that maximizes the biomass for just a single organism at the expense of everybody else. And this is, so Additionally, CTFBA allows for suboptimal community biomass, which again helps balance the trade-off between individual growth rates and that of the community. Great. So cooperative trade-off FBA allows us to treat metagenome allows us to treat metagenome scale models with the same methods as genome scale metabolic models. For example, PFBA, minimal media, etc. And to this point, we focused on making genome scale models, but to make the metabolic models of the microbiome community or metagenome scale community level metabolic models, we'll be using MyCom, which extends flux balance analysis into microbial communities and leverages some of the approaches I just highlighted, such as the cooperative trade-off flux balance analysis and parsimonious FBA. To initially build the models, we need to pass in the relative abundance of bacteria in the sample, which we did in the notebook. Due to sequencing efficiency differences, there may be some bias towards some bacteria over others, but the abundance from sequencing data should be more or less representative of the community. MyCom will then map this abundance data to a database containing hundreds of genome-scale metabolic models of common gut microbes. MyCom then uses the reconstructions of those taxa present in the sample to build a massive stoichiometric matrix, like the one we discussed earlier, that includes not only the internal reactions within each taxon, but also the exchanges between them and the external reactions, which in this case is the host. We can then specify a diet that is representative of the food being eaten by the subject of the model and use FBA with an additional regularization step to calculate unique growth rates for all the bacteria based on the processes we've outlined previously. Following this, estimates of all the fluxes in the system can be generated based on those growth rates, returning the most likely flux distribution. And this is where we can leverage techniques like the parsimonious FBA or minimal media. This gives us a huge output of data, which we can then use to make detailed and interesting predictions about imports, exports, and the inner machinations of bacterial reaction networks, as well as codependencies between these bacteria or possible interactions, and a testing ground for potential interventions. But does it work? That's the age-old question. Here we see results from the original MyCom publication validating the cooperative trade-off FBA approach described. On the left, we see distributions of individual taxa across a large number of communities and conditions. What we see is that if we don't integrate a regularization step, that is, no L2 parameter minimization, there's a very small number of taxa that can achieve high growth rates. The majority effectively are not growing at all. You can see that here in the distribution for the no L2 case. Obviously, this is not what's actually happening in the microbiome, and the solution is not relevant to the system. If we apply the L2 regularization strategy and require the community to achieve its theoretical maximum growth rate, we see that a lot more taxa are able to grow. That is, the L2 regularization better distributes growth across the individual community members. However, as previously noted, this maximal growth rate might not be achievable with all taxa growing. Lowering the trade-off value just a little, maybe to 0.7, for example, which is allowing the community to achieve only 70% of its theoretical maximum, we see that almost all the taxa are able to grow, echoing what we expect to see in the microbial community. That is, that if we measure the microbe, that it's likely present. The team that was a part of this study also validated predicted growth rates by comparing them with replication rate, which is generally proportional with growth rate. To be able to infer replication rate, the team had metagenomes. And in general, it's been shown that coverage profiles of metagenomic reads for fast-growing bacteria can be used as a proxy for growth rate. You can do this by measuring the decay and read coverage from the origin of replication, as noted in the middle figure. Using this method, the team could estimate growth rates in vitro and compared these to growth rates predicted by MyCom. You can see in the panel on the right that without regularization or at high trade-off values with regularization, the predicted growth rate did not correlate very well with calculated growth rates. Lowering the trade-off value slightly showed a much stronger correlation with calculated growth rates, thus showing that CTFBA provides a biologically meaningful solution to the community growth rate problem. So, in short, cooperative trade-off with L2 regularization helps us reach a solution wherein most taxa are able to grow and provides us with biologically meaningful growth rates. Awesome. These methods seem great and the math all checks out. So why is it taking so long to run these workflows? I can tell you it's probably still running. Well, typically MyCom models are a bit more complex than the examples I've been using to illustrate the methods, like our 2 E. coli community. This is a visualization of a standard metagenome scale metabolic model. Each pink dot represents a metabolite and each blue dot a reaction, with black lines connecting them. Each of these regions is a single taxon in the community, where you can see these sort of like clusters being formed. You can see that the community model contains nearly 70,000 reactions, over 45,000 metabolites, and nearly 300,000 connections. So you can imagine that finding the optimal flux solution in this model takes some computational power and sometimes a little patience. I know at this point you must be excited to see the results of our MyCom growth simulation, and your wait is almost over. But first, let's take a look at some of the visualizations we'll create the output from our results. Since MyCom gives us so much great data, it can be a little overwhelming at first, but don't worry. It's actually very easy to make great visualizations using built-in MyCom functions, or extract the data to use in using your own favorite visualization package. First and foremost, we'll take a look at growth rates of the individual taxa in each of our models. MyCom has a built-in function for this, so it'll be very easy to build this particular graphic. Here we have a scatter plot with log-scaled growth rates on the y-axis for each taxa on the x-axis. This plot is a great way to check that your models are growing correctly at a typical growth rate, like what we see here. It can also tell you which microbes are doing well and if high growth rates in the dietary context you have used to simulate the models. For instance, here we see that Prevotella has a high growth rate and seems to be thriving, as it's the furthest to the right on this plot. We'll see in our results that the growth rates for individual taxa can differ quite a bit, and they also might differ between the results that I show and the results that you generate. Again, due to some of the limitations of using Google Colab for our model simulation. This echoes one of our primary points, which is that context is a major factor in the metabolic activity of the microbiome, and it's very important to consider. Next, we'll look at a visualization of the niche space of each taxon in each sample. The niche space represents the context-dependent way in which a taxon uses its environment. In this case, how it uses the metabolites available to it. So we can look at, for instance, import fluxes or export fluxes. Since plotting all the metabolic exchanges wouldn't be possible, we'll use TSNE or TSNE to make the plot. This technique takes high-dimensional data and reduces it to 2 dimensions, such that similar points are closer together and less similar points are spaced further apart. This is akin to principal component analysis, if that's something you're familiar with, or PCA, but is the nonlinear version. In addition to TSNE, there's also a UMAP, which was used to generate the figure shown here. This type of figure can give us an idea of the metabolic niche each taxon falls into and how similar or different it is from the other taxa in the community. Like I mentioned, niche space is context dependent. Some taxa change their metabolic behavior depending on what metabolites are present in their environment, allowing them to survive in multiple niche spaces. This adaptation can be helpful for their survival. In the example niche plot shown, I've used UMAP to accomplish the dimensionality reduction, which is fairly similar to t-SNE conceptually. Here I'm showing the results for individual taxa across thousands of samples. Interestingly, what we see is that the individual taxa often cluster into one or just a few discrete locations, suggesting that they often select from a finite number of metabolic strategies. Finally, we'll take a look at the metabolic imports predicted from the growth of each metabolic model. As shown here, we'll construct a heat map illustrating the amount of different metabolites produced and exported from taxa in each sample. In this case, each row represents a metabolite, and each column represents one of our samples, with cells colored in a logarithmic color map. While MyCom does have a built-in tool to build heat maps like the one shown here, we'll use this opportunity to take our output and build a visualization in another package. In this case, we'll be using Seaborn. This will additionally allow us to plot the results of growth on both diets or different disease contexts. Similar to the niche space, the metabolic activity of any taxon in a community, and therefore its exchanges, is dependent on the context in which it grows. In this particular heat map, we can see how for individual taxon, it represents its different growth niches as indicated by the clustering at the top, where we have a high, moderate, and low growth modes. And you can see that those different growth modes are associated with different metabolic import fluxes, where in the high growth rate scenario, the organism is importing a lot of things. And then in the moderate cluster, you see it sort of diversifies its strategy and is importing as much as it can, but not everything. And obviously, when it doesn't grow, it doesn't consume very much. Great. Well, we've made it through the theory. So now let's jump back to our notebook and see that whether or not our results are done, which I think by now they probably are. Great. So here's the medium. It does look like our models were built and it took about 11 minutes. And also, it looks like we got our growth results. Great. So if for whatever reason one of these steps didn't work for you, or it's still running and you don't want to wait, or maybe you're just now joining us, you can import a pre-computed growth result from the treasure chest using this cell. So growth rates are pretty straightforward. Let's take a look at what our growth rate results look like. Here we have a table of our growth rates. You can see the different abundance, the growth rate, some details about the model for each of our genera in each of our samples. There's also some details about the modeling. Here I chose to use a trade-off of 0.8, which is allowing the community to achieve about 80% of its theoretical maximum biomass. And then, of course, we have all our metadata. Also, we can look at exchange fluxes. And, if there are metabolites that we're particularly interested in, we can focus on those. As you might have guessed from the slides, one class of metabolites that we might be interested in are the short-chain fatty acids. As an example of that, we can look at the SCFA butyrate, and we can see that the reaction associated with butyrate is this one. We can see that this is its name that's used to represent the metabolite, as well as its more common name. And, there's more information here that you can look at. Great. Now let's build our first visualization. Okay. You can see it showed up here in my materials folder. And I'm going to download it. And let's see, can I see my downloads? Oh, it's still downloading. You can also try clicking on it, but there we go. So here's our growth rate plot. You can see relative to the one that I showed in the slides, the axes here are flipped, but it's still showing the same thing. You were looking at the logarithmic scale growth rates, and then it's ordered by the who's growing the fastest. And you can see, for instance, Prevotella again seems to be one of the fastest growing strains, or genera in this case. We can also see, we look at a few others like the Bacteroides phocacola, or sorry, phocacola, not Bacteroides. And you can see, for instance, for this guy, as well as for Bacteroides, they also achieve pretty high growth rates, which is expected given that they tend to be dominant genera in the microbiome. Bacteroides is often between like 50 or 60% of the community. And you can see here, actually, there's like quite a wide distribution in the growth rates for these guys across our different samples. The same is true for this Faecalibacterium, which is one of the common butyrate producers in the gut microbiome. Cool. So now I can go back to our notebook. And yeah, I just mentioned Faecalibacterium. So let's take a look at that guy specifically and see how its predicted behavior might vary across our samples and more specifically across disease contexts. So here you can see I've extracted the growth rates associated with the taxon Faecalibacterium. Now I have a table of just those growth rates across our 10 samples. And I can try to make a box plot. And interestingly, we see that the growth rate of Faecalibacterium here seems to be predicted to be higher in those with Parkinson's disease than without. Granted, this is a very small sample size, and these are probably not statistically significant. I'm actually not going to try to test it, given the small sample size and the variation here. Additionally, you might find that your results vary a little bit from mine, but let's dig into this a little bit further. What about other genera? Let's look at some of those guys. So here I'm extracting growth rates from the top 10 genera here. And what we see is the growth rates now being compared across disease contexts. And again, we're seeing some differences, it seems like. And again, those differences might vary between what I'm showing and what you generate, just because of the nature of this process and how challenging it can be to get a one solution using the kind of limitations we have with the Google Colab. But yeah, again, we're seeing some interesting differences. Again, a lot of variance here. You see there's a wide range of values in these samples. In some cases, there's growth rates for some of these guys are only showing up in a few samples, so you can't even really make a box plot. But nonetheless, yeah, we see some differences, and maybe they're what we are expecting, maybe they're not. But to know more, we're going to have to dig a little deeper. So next, I mentioned the growth niche. So we can use this built-in micom function to look at the import niche of our taxa. And this will produce a HTML output similar to the previous one. This one's called niche.html. So I'm going to download it so we can look at a preview. And for mine, you can see here, this one is using Tisney. And you can actually look at the individual genera. And if you click it, it just shows that genera. And so, for instance, for Ruminococcus, it looks like it has it was only predicted to grow in maybe one of the samples. So it's only showing 1 point. But let's see if we can find that Faecalibacterium. So Faecalibacterium seems to be like a light gray, so maybe this one. Yep. And yeah, as the box plots sort of indicated, there does seem to be some variation here. You can see, for instance, this one point is very far from the other points. And, you might argue that these points are forming one group. You could also maybe argue that there are maybe subgroups within these points. Maybe you might argue that there's maybe 2 groups here, and if you count this one, maybe 3. So it's possible that it has multiple strategies that it's leveraging across these samples. And yeah, maybe let's look at one more. How about this guy? Here we have Bacteroides. And again, yeah, seems to be a kind of a big spread here. Again, suggesting there's kind of a large amount of variation in the metabolic strategies, in this case, the things that are being consumed by this genus across our samples. And this might be due to intrinsic differences in the samples based on the composition of the community. It could also be due to just variance in the model simulation. Again, we're gonna have to keep digging to find out more. So those are great. Now we might want to compare fluxes across these samples to try to get a better idea of what some of the functional characteristics are and how they might differ across these disease contexts of Parkinson's disease. So yeah, to keep with the theme here, let's continue to look at butyrate and see if there are any differences in the production of butyrate across different genera. You can see there's some different transformation steps that are needed here. I'm looking at the butyrate production, so I need to look at the export fluxes. So to do that, I'm first getting the exchanges, as they're called, the metabolites that are being consumed and produced by the models, the community. Then specifically getting the export fluxes. Then from those export fluxes, extracting butyrate. I then log transform it, as this makes the data a little bit closer to normally distributed. And I have to add a pseudo count to the flux just in case there are any zeros. And then from there, I'm extracting the top 3 genera with the highest butyrate flux. In this case, it looks like it's mostly Faecalibacterium that is producing butyrate. And there's a few isolated examples of other butyrate producers. Again, this might be different for you guys, but in general, I've seen through multiple iterations of this process that Faecalibacterium tends to be the one producing butyrate most frequently, though it might not have the highest flux for you. So the box plot is one way to visualize this. We can also use Seaborn to make a heat map. Sorry, that's the next step. This is actually breaking down our box plot butyrate production into the different disease states. And in this case, what we're seeing is that while Faecalibacterium grows higher, or is predicted to grow higher in the people with Parkinson's disease, you see, in my case, that the butyrate production is predicted to be much higher in those without Parkinson's disease, which does seem to be in line with the idea that maybe those without disease have a lower inflammatory state in their microbiome. But again, this might be variable for you, and we will continue to dig a little deeper to see how this looks. So we looked at the butyrate, as a box plot broken down by disease status. We can also visualize it as a heat map, as I was just mentioning. This gives a better way to look at butyrate production or the production of any metabolite across the individual samples instead of showing sort of more of like the average and variation. Again, we see that Faecalibacterium is mostly the only genera that is producing it in our samples. And here you can see the coloring here is trying to show you the disease status for the Parkinson's disease, where one color is representing yes and the other is no. Unfortunately, I forgot to put a legend here, but because we know in my case that the butyrate is higher in the individual without Parkinson's disease, you can see the highest values are associated with this blue color. So for me, blue is the not people without Parkinson's disease. And you can see that they do seem to form a cluster right here in the middle, which is interesting. It's suggesting that in my case, there is some similar behavior here. And the people with Parkinson's disease are kind of on the edges. But that's just for the case of butyrate. Next, we can look at the overall butyrate production. And we can see that in my case, the butyrate production overall is much higher in those without Parkinson's disease, again, due to the activity of Faecalibacterium. And this is in line with the results that we've seen yesterday, generated by Alyssa, that showed that there was some differential abundance of some of these butyrate producers across these disease states. Again, this result might vary somewhat. I've found that in previous runs with this, I've generally seen that the difference is consistent, but the magnitude might vary. Your separation here might not be as great as mine. So yeah, this is looking good. We are seeing production of butyrate, and there does seem to be some difference across our disease status, though I should note that the overall value here is still fairly low, and that's because we assumed that everyone here is on a Western style diet, and that was because we didn't really know very much about their dietary information, which is one limitation of the study that we leveraged. We found in other situations, if you use a representation of like a high fiber diet, you can actually get much higher fluxes of these short chain fatty acids like butyrate. So although we are seeing a difference, the overall magnitude here is still somewhat low. And that speaks to the importance of having dietary information when you're performing these microbiome studies, because that is such a strong driver of variance, and it's very important when we want to do these types of modeling techniques. So that's great. We got to zoom in on this one molecule and look at its differences. Now let's look at a more broader perspective of the production fluxes. What are the things that the microbiome is producing? So here again, I'm extracting the fluxes, the export fluxes, in this case, using this production rates function. I'm then merging that with the metadata and turning it into a wide table, as you can see here, where I specified what I want the index and columns to be. Ultimately, from what I did here to what you're seeing, there has been a transpose of the table. But importantly here, yeah, we have both the disease status and the sample ID as one unit of information. And then the other unit of information is the name of the metabolite associated with each flux. And then the values are our log transformed fluxes. Again, adding a pseudo count just so that if there are any zeros that are handled. You could consider other approaches. For instance, Alyssa yesterday showed a great approach where she filtered out the genera that were very sparse after doing this log transformation. Here, I have not done that. So we do have to be mindful that there are values here that are effectively zero. For instance, these negative 6.907 numbers are all essentially zero. Great. So now we have our flux table, and we can build another one of those heat maps. And here we have a heat map where I've selected the fluxes with the, first I've sorted the fluxes, and then taking just the top 25 with the highest average magnitude. And you can see those. And here we can see there does seem to be a sort of interesting pattern in the heat map with this sort of like diagonal behavior where each sample seems to have a high export of just a subset of these 25 metabolites. However, we see up here in the clustering of the samples based on the disease status, these flux patterns, these global flux patterns, don't necessarily capture very much difference in disease status, which might suggest in this case that while we do see some interesting differences in like the production of butyrate, the global export patterns themselves are not necessarily very indicative of whether or not someone has Parkinson's disease. And this might be because there's other features here that we haven't taken into consideration that are driving this variation. Again, who is present in the community could be a factor. Diet here again is not well represented, so it's hard to know what is maybe driving this variation, or if it's just variability in our simulation results because we're not using the solvers that we'd like, and our sample size is small. So, take this all with a bit of a grain of salt. It is very interesting that we can do this, but we would need more samples and more data, more information to really dig into this and understand what's going on here. Great. So now that we've gotten to this point, now we're going to turn it over to you guys. So I've put together a little exercise here for you. And what I'd like you to do is to try to make one of these heat maps, but instead of this is looking at the community level export flux. Let's now instead look at the import flux for an individual taxon. So here we can look at the growth rates for the top 10 genera, and we're counting like their presence across the samples. And you can see Bacteroides is present in nearly every sample, as is Phoca-Cola and Faecalibacterium. But there's a few other genera that are present in at least half of the samples. So this might be a list that you can pick from. You definitely want to pick something that's more present in more than just one sample to be able to make a heat map. I think you'd probably want to pick one with 4 or more that's present in 4 or more of the samples. And this I've given you sort of a skeleton here to get you started. Please look at earlier steps in the notebook to try to inform you how to proceed here. You'll want to get the exchange fluxes, then get the specific exchanges for the genus you're interested in, then get the imports and do a few transformations, like adding the metadata, making it a square table instead of a long table. Do the log transformation, etc. And then once you have that table, then in this next cell, you can try to make the heat map, much in the same way that I made this heat map. Yeah, so with that, I will just briefly jump back over to the slides and take a short break so that you guys can work on that. So here's your slide to keep you motivated maybe. And yeah, please, if you have any questions, hop on the Slack and ask. And in the meantime, I will be back. All right. Welcome back, everybody. So yeah, hopefully you were able to figure out this exercise that I left you with. But if not, no worries. We will walk through it together. So let's see, I'm back here in the notebook. And yeah, it looks like I'm still connected, which is good. So yeah, let's walk through how we can get this solution. So first things first, we want to get the exchanges. So here, what I'm going to do is I'm going to make a new code cell because I tend to like to go one step at a time and visualize my results as we go so that I can feel confident that it is correct. Oops. So first things first, we want the exchanges. So we can use growth.exchanges. You can see if I run that, this is what it looks like. So this is the first step. I'm going to copy that, put it right here. So that'll give us our exchanges. Next, we want the exchanges for a specific genus. I'm going to choose the Caelibacterium. So I will copy that. And now you might have been able to look at some of the other code cells to try to infer how you do it this step. But in case it wasn't clear, let's see. Here's our exchanges. Or actually, I haven't saved that yet. So let's say exchanges equals growth dot exchanges. And now we want to get a specific taxon. And so the way we do that with a pandas data frame is you take the data frame. And we didn't really explain how to work with pandas, but typically with a data frame, if you want to select a column, you use the brackets, and then I can say taxon. So for instance, that is going to be all our taxa. The medium is representing like the kind of bulk properties of the community. So if we want a specific taxon, what we can do with this table is take our column of taxa and then use a logic comparison to see if it equals the taxon we're interested in. So I'll do like that. Now, if I run this, you can see it becomes a true or false table. And then we can use that true or false table to subset our larger exchanges table. So if I put that true or false table inside of the larger table, now we only get the exchanges where the taxon name is equal to Faecalibacterium. So that is the solution to the second step. So now we have genus exchanges. Now we want to look at the imports specifically. And as you could maybe guess, we can do that exactly the same way as how I selected the specific taxon. We can select the specific direction of import or export. So to do that, I'm going to use the direction column. See, oops. Let's see. Oh, I spelled something wrong. Great. So I'm gonna use the direction column, and yeah, same as before. I'll say I want the direction that is equal to import. And then I will put that all inside of itself. Great. Now we have all the exchanges that are in the import direction and also for Faecalibacterium. So that is the solution for that step. Also, just as a side note, I chose to do it stepwise as I'm showing you now, but alternatively, you can use the same approach and do it all in one go. So I could have done instead this. When you have more than one thing, you have to enclose each comparison in parentheses like I'm doing. And then you need to add like a logic operation. So if we wanted the things to be all Fecalium bacterium and all imports, I could do this. And then now it's saying, yeah, I want all my things to be Fecalium bacterium and also to be import. And then I can say that equals genus import. And then I can actually just get rid of this. And then I spelled it wrong. Great. So those are two equivalent ways to do it, either like this stepwise or all in one go. Now we want to add the metadata. And if you remember, this is something that has been done a few times, both in this notebook and from yesterday. And so to add the metadata, we'll want to use the pandas function merge. And so, and what we're going to want to merge on is something that both the metadata and our current table share, which if we look at the metadata table, you can see they both have the sample ID. So that is what we will merge on. And so to do the merge, it's used pandas.merge. And we'll take our genus imports table and our metadata table, and then we'll merge it on sample underscore ID. And we can look at the results, and you can see it seems to have correctly merged. And great. So I would say genus import equals that. And I will also copy that here, put it here. Great. So far we're doing pretty good. We got our genus import table with the metadata. Now we want it to be in a different format because to make a heat map, the heat map visualization is not going to work so good if we feed it this table. This is great for other seaborne functions like boxplot, because for that, it actually prefers it be in this format, which is like a long format, where you can see each column is a different feature of the data, and each row is just like a different index for that feature or different value. And but for the heat map, we actually want it to be more like a square table, maybe something you would maybe more traditionally be familiar with when like you're importing data and stuff, where maybe rows are samples and columns are fluxes, or vice versa. And so to do that, we are going to want to use a function called pivot table. So to do that, we will use, you can see, for instance, another cool thing about these notebooks is if I have an object, in this case, a pandas data frame, and I do the dot, like I'm going to use a function associated with that object, and I press the dot, and you can see it brought up all these options. So these are like some of the things, some of the built-in pandas functions. And so if I put P, I, V, you can see it brings up a couple of our options. And here's pivot table. Also, for those that maybe aren't super familiar with Python or pandas specifically, if you ever have like a question about a function, obviously you can Google it, but also you can always use the question mark. So if I use the question mark here, you can see it brings up like all this documentation on how to use pivot table. It's maybe not super easy to read just because in this like text format. So finding the like web page might be a little easier to look at, but help is there if you want it. Okay, so we want to pivot the table, and we're gonna do essentially a very similar thing to what we did here. So yeah, I think I will actually maybe just copy this. Okay, now we're gonna go here. Pivot table, great. And let's see, columns. Let's take a quick look at our table again. Okay, so in this case, we probably want the column to be not name, but reaction. Values are still going to be flux. And I think that's all we need. So if I do that, oh, I see. Typo. So I could actually just, I did it a different way above, but this is another equivalent way to do it. Alternatively, you could do it the way I did it above and use pd.pivot table, then feed it genus import. Both are valid. Let's see. So that's how we pivot the table. So I'm going to copy this. Great. Now this part is actually just converting, it's filling the zeros or the nans actually. So you can see there's a lot of nans, and that's because, for instance, for this flux, you can see that Faecalibacterium is only predicted to import this molecule in one sample. And all the other samples, it was not necessary at least in the results that I got. So this function here is pivoting, or sorry, it's transposing and then filling the nans with zeros and also taking the absolute value. Let's see. Oh, I did something bad. Let's see. I think I might fill them. It doesn't like the fact that we have a string here, which is surprising because, oh, I see. I see what I did wrong. I forgot to save this as a new thing. So I was trying to take the absolute value of some of our metadata, which some of it is not numerical, which doesn't make any sense. There we go. So now you can see the zeros. The nans have been filled with 0, and it is now in a format that is nice. Great. So as an optional thing, you don't have to do this to get it to work, to make the heat map. You could actually just make a heat map with what we have now, but there are still a few more things that would be nice to do. One of those things is getting the reactions, or like the names for the reactions, because you can see right now the format that is used is a little bit obtuse. Like it's hard to know what some of these things are. And so to do that, we're going to get the annotations. Again, we're going to go back to this growth results, and we're going to use growth.annotations. If we look at that, we can see that this is a table with the index equal to, that is using the reaction name, and then it has all this other information. We looked at this a little bit earlier when we were looking at the case of butyrate. So if we want to get our annotations, we can do that. So I'll say annote equals that. Can also copy this, put it here. Now, let's see, let's look at annote. So you can see that the index is the reaction. Here, that was sort of the intent of this line, but we don't actually need it anymore. So now we want to get the names. And so this line actually accomplishes that. So I tried to throw you a bone there because that was maybe a little bit complicated. But if we want to understand a little bit what's happening, we can look at the different parts of this function. And so if you do this, you can see annote.name gives you the name column. Again, with pandas and with a lot of this stuff, there's many equivalent ways to do the same thing. So I could also do it that way. So once you have the name column, which is ultimately what we want to convert these reactions into names. So to do that, I'm going to convert this into a dictionary. So I'll use the function toDict. And now you can see we have a dictionary, and this dictionary gives us a mapping between this reaction identifier and the name of the reaction, or the metabolite associated with the reaction. And so then what we want to do is use that with the mapping function. So if we again look here, our index is these reaction identifiers. So then we use dot map, and then we take this and we put it into the mapping function. So the mapping function is taking whatever it is you're mapping, in this case, the index, but it could also be a column or something else. And then it maps, and to map, it needs a dictionary that will take these values and convert them into something else, which is what we have here. So I'll get rid of this cell. So if I do that, you can see it transforms those index values into more meaningful names. And then you save that as the new index, and then your import table will look like this. We're getting pretty close. So now we want to do the log transform. So to do that, we will use numpy.log. You can also use, I just use log, but you can also use log 10 if you have a preference for that. So then we're going to give it genus imports. Again, you see there's a lot of zeros here. So I will have to add a pseudo count to be able to log transform our data. You could also look at it not log transform, but you should know that these fluxes are not normally distributed in their linear form, which is why we log transform them. Also, you can see I'm adding a very small count, which is, smaller than most of the values here and close to 0. I wouldn't want to add like 1 because that is much larger than some of the values. So when adding this pseudo count, you have to be careful with how, considering the impact. Another option would be to just look at the fluxes where there aren't any zeros, but that might drastically limit what we're looking at. Okay, so if I do that, you can see now we have log transformed values. And also, let's see, I'm going to actually get rid of that for a second. We can just very quickly, just to drive home this point about the log transform, if we want to look at a specific metabolite, let's just pick L-cysteine. You can see its values, and you can even just get the values. This maybe isn't the best illustration just because we have so few samples. But if you had more samples, what could be informative here is to make a histogram of those values. And you can see, like I said before, they aren't very normally distributed. And then if I add my pseudo count and log transform, we can look at the effect that has. And you can see actually it's still, in this case, fairly binary because the pseudo count is all 0. We can also look and see like what does it look like for the values that are not close to 0. And again, pretty sparse. If we had more values, you would start to maybe see the normal distribution. But in this case, we don't have enough. But anyways, the point there is just to say that the log transform is needed to make these values more normally distributed. Okay, so now let's see. Let's copy this little bit here. And now we might want to sort our fluxes by the most highest variance, because we might be interested in the ones that are most different across our samples, which would maybe give us some insights about what's going on across the disease context. So if I go back here, one way to do that is to look at the variance. And so to do that, we can use the variance function, so .var. So if I do .variance, you can see it's actually kind of doing a funny thing just like this, because the function will either go row-wise or column-wise. In this case, you see it's showing just the total variance across all metabolites in a sample. That doesn't make a lot of sense. So we need to actually tell it to go and calculate the variance row-wise instead of column-wise. So to do that, you can give it this axis argument. The one we just did was 0. And if you give it 1, now you can see we have the variance of each metabolite across our samples. So now that we have the variance, we can sort the variance values. So we do the dot sort values. And so as you're starting to see, one cool thing you can do with these pandas data frames is you can sort of iteratively add like layers of function. So first I did the variance function that gave us a new data frame. And now I'm doing the dot sort values on that data frame. And so if I do that, it actually sorts it from lowest to highest. So you see the variance of water and carbon dioxide are the lowest. And that's because those are essential metabolites that are probably imported very similarly across all our samples. In this case, it's saying they're exactly the same. So now I'm going to say ascending equals false, and that will flip the order. It'll go from highest variance to lowest now. And now if I want to use this order for our data frame, I'm actually going to use the index, which is the same index as our data frame. So I'll say that as a new variable. I'll call it index. And then to sort our data frame using that index, I will just feed it into I will select using the index. So that's why I'm using .loc. It's like .location, where you give it the index and the columns. So I just give the index. And now you see the order matches the order that was obtained by sorting on the variance. Great. So there's that. There's that. And voila. Now it's the same as it is here. Great. So yeah, I don't really need this code cell anymore. Okay, so now we have our table. And now we want to make a heat map. See, I'm getting rid of these extra cells. So to make that heat map, we will basically want to just use this same snippet of code here. Maybe make a few tweaks just in case there's something different, but let's just try it and see if it works. I'm going to put that here. One key difference is this. Also, one thing you might want to do here, if I run this again. Let's see, let's run this one. Now we can just look at df here. So this is the format that we will want our table to look like. And let's see if we can achieve that here. it looks pretty similar. So this will probably just work. Let's see. It didn't like something though. And that is because, I see. Because I forgot to update this part. So here we're using genus imports again. And voila. It seems to have worked. Let's just double check. So df equals genus imports. Then we're going through and creating a color palette based on the Parkinson's disease column in df. We're then creating this color spectrum here based on the samples. Let's just double check here. Great. And then, yeah, this looks good. And it's interesting too, because it does seem like, oh yeah, I see. I was like, it looks the exact same as the other one, but that makes sense. Let's see. Yes, I see what I did wrong. This shouldn't be DF. This should actually be GSMX. So yeah, this can kind of give you a flavor of, sometimes coding is trial and error. At least it is for me. So you can see we managed to figure this out. And here, yeah, now we have the heat map for our Faecalibacterium fluxes. And yeah, one interesting thing you might notice is that compared to the global patterns of the community, here for Faecalibacterium specifically, you do see a little bit more separation between the disease status and its import flux patterns. You can see most of the of this feature here are grouping together. Again, no legend, but let's see if we can find, let's see if butyrate shows up because that might give us a hint. I don't see it, so let's maybe make this a little bit bigger. What if we do 50? Also, this clustering might change. Yeah, you can see the clustering will change a little bit. Still, you're getting some grouping, but there's obviously some fluxes that are not that different between the two conditions. And yeah, doesn't look like butyrate is showing up too much, or at least I'm not seeing it. It's possible it's not labeling. Yeah, it might not be. No, it does look like it's labeling every row. Maybe it's there, I'm not seeing it, but also, like I said before, the magnitude of that particular flux was kind of low from what we've seen, even though there was a significant difference. Regardless, though, yeah, we can see that we can kind of dig into this Faecalibacterium import flux patterns, and you can see for some things there is sort of distinct patterns across our disease status, and for other fluxes, not so much. I think is a little easier to look at with fewer features. Um, yeah. So yeah, for instance, you can see for formate seems to be pretty high import across all the conditions, whereas so you can look at succinate, and succinate seems to really only be highly imported in this one condition. And these, to really understand why that is, we'd need to dig a little bit further, but you might hypothesize that this could be due to differences in community composition. Maybe in this sample, there's less competition for succinate, or maybe another organism is producing succinate that is being exchanged with Faecalibacterium in this case. Again, you'd have to dig a little deeper to know, but this gives you a flavor of how you can sort of dig through this data and pull out this type of information. Great. And so just as a final note here, one thing that we have to be very mindful of is the parameters that we're using for our simulations. Um, I provided some defaults that we used for those simulations. One of those parameters is the trade-off value. This trade-off value, as you might remember, is the fraction of the optimal community mass that we're allowing our models to achieve. So we use 0.8, so we let the models achieve as low as 80% of the optimal solution, because we know that in high diversity communities, you're generally not going to achieve that optimal biomass with everyone growing. So choosing the right value is very important. And to inform that choice, you can you can do the analysis that's presented here. If you try to run this, it will actually take quite a while because it has to do the growth simulation many times. But you can see here's an example of what it could look like. And here we're showing the fraction of growing taxa, those with, non-zero growth rates. And you can see how that looks across these different trade-off values. And you can see if we don't use a trade-off value, so no regularization, you can see that very few taxa grow, as was alluded to earlier. We're seeing only about 10% or less across the samples that are shown here. And as you increase that trade-off value, or sorry, as you add the trade-off and then decrease it, allowing a more permissive growth solution, you can see there is sometimes a sort of sigmoidal shape, or in this case, maybe more of a hyperbolic shape. And generally, we want to choose the value at which you get a majority of the community growing, so maybe somewhere between 70 and 80%. Which is why we chose this 0.8 value. So you can see right when it starts to level off. And that's just based on empirical measurements showing that around 70% of the cells in stool are still viable when they do flow cytometry. But if you have more data, or you say you have like metagenomics data, for instance, you could do the analysis that looks at the inferred replication rate and then correlate that with the growth rates across these trade-off values, and then choose your trade-off based off of that. So again, you have to, it's important to consider these parameter choices when you're doing these simulations, so you can get more realistic results. Great. And so we still have a little bit of time left. So I think with the remaining time, it might be nice to actually show you guys the application of these tools in another context where we're able to do more validation and have a more tangible application. So I'm going to actually go ahead and pull up a manuscript that was published recently by Nick Quimboman in our lab that shows how you can use these microbial community scale metabolic modeling techniques to predict personalized short chain fatty acid production profiles in the human gut. As you can see, this was recently published in June in Nature Microbiology. And I think provides a great example of how you can use these techniques to better understand SCFA production. So I will leave it to you guys if you're interested to try to read the paper if you're interested. I just want to kind of highlight some of these figures that show some of the results that Nick generated. So this one is a nice overview, probably a nice refresher from the stuff that I was showing earlier. So yeah, what Nick did is he was able to construct different growth media representing the Western diet as well as high fiber diets and some other interventions um, based on what people could be eating. And so with that growth medium, he used MyCom to build and do the growth simulations, um, much in the same way that I showed today. Um, but also he was able to validate the predictions of the model using both in vitro and ex vivo samples where butyrate, propionate, and other scFAs were measured, um, so that we could compare the predicted fluxes with the measured fluxes. So to do that, he looked at scFA concentrations over time and got the fluxes, and then was able to look at the predicted versus measured fluxes. And so when he did that, one of the first tests leveraged in vitro cultures. This is data from a collaborator of ours, Ophelia Venturelli's lab, who does a lot of work looking at in vitro cultures and ways of like modeling them to be able to understand the function of these communities in vivo. And so the data that Nick leveraged had communities of various composition that fell into low and high diversity regimes, low being 1 to 5 taxa and high being 10 to 25. And when looking at the measured butyrate production in these communities, one of the things Nick found is that in the setting with low diversity, the prediction actually is not, it's correlated with the measured butyrate, but you can see the prediction is not very strong. Um, which, whereas in the setting with high diversity, you see the prediction, it actually does match the measured butyrate concentration fairly well. And this actually speaks to one of the limitations with this modeling framework that we've found empirically is that at least using the community metabolic modeling framework, MyCom, we find that in low diversity settings where we have only 1 to maybe 5 taxa in this case, generally the predictions tend to not be as good and you can see higher variance. And they stabilize generally with higher diversity, which is an interesting feature that is likely related to the L2 regularization that is performed. Additionally, when using ex vivo culture, which is taking stool samples from patients and then looking at the chemistry of those stool samples over time within a window of time where they're still biologically active, what Nick found is that in that setting, looking at these 3 scFAs, you can see that both butyrate and propionate are pretty well predicted, and this is actually across a few ex vivo studies, whereas acetate is not so much, and that actually makes sense because acetate is actually a really core metabolite in the greater cell metabolism. It is also a product of overflow metabolism. Thus, it makes sense given its like complicated role that predicting its production might not be so great. And then, just breaking those results down a little further, you can see, how the ex vivo results look across these 4 studies and across these different scFAs. Again, you see that butyrate and propionate are pretty well predicted across these different studies. The previous figure was just showing the aggregation of all of these samples. And you can see that like the slopes are fairly consistent, which is nice validation. And then so that shows that this modeling framework does have some utility. And a cool application of this is to then try to use it to better understand inflammatory states and their relation to SCFAs in human cohorts. And so to do that, he leveraged this study from the Segal lab, which looked at a population and gave them all a 10-week high fiber dietary intervention. And one of the things that original study found was that while, generally speaking, eating more fiber is thought to be something that we can all benefit from, they found that there was actually fairly variable response to this intervention, with some people falling into low or moderate inflammation groups, and some people actually having an adverse reaction to this intervention and responding with a high inflammation state, suggesting that your microbiome's response to fiber can be very variable and really depends likely on your immune status prior to the intervention, as well as like who's there. If your microbiome is not well equipped to handle the fiber, it can, potentially be a problem, as this study shows. And what's cool is using MyCom to predict butyrate production in this study. Nick found that for propionate in the inflammation group here, the lowest inflammation group, you actually see a positive relationship with the time point in the study and the production of propionate, which makes sense. As you're consuming more fiber, maybe you're producing more propionate, and that is what's leading to this lower inflammation state. Again, yeah, you see on average, the propionate concentration predicted is higher in the low inflammation states than it is for the high inflammation state, which is maybe providing a mechanism for why that occurs. You see in the high inflammation state, actually, there's a negative association between propionate produced and time. And finally, I'm actually gonna skip over, oh, actually, no, I'll talk about this one. Yeah, another thing is that the model predicts in another cohort of very large cohort, the Airvale population that Alyssa alluded to yesterday. When you do the same prediction between using MyCom to simulate the SCFA production, and you look at the correlation with this prediction and the measured blood chemistries and metabolites, you could actually see that the model predicted butyrate is strongly associated with a number of clinical markers of inflammation. And with the right sign, you can see things that are indicating higher inflammation state are generally anti-correlated with the predicted butyrate concentration or flux, whereas the actual measured butyrate concentration doesn't really show that same relationship. Again, kind of suggesting, as we implied earlier, that when you try to measure the concentration of these metabolites in the body, sometimes a single concentration value is not very informative because it's actively being utilized by the body, so it's not necessarily going to sit around and accumulate. Additionally, we see some association between like BMI and predicted butyrate, as well as propionate, suggesting that if your microbiome is predicted to produce more butyrate, you're generally going to have a lower BMI and lower markers of inflammation, which are in line with the mechanisms that butyrate is thought to, and propionate are thought to leverage in the body. And then finally, using the same population, Nick was able to show that if you subject these different people to different combinations of like a European Western style diet or high fiber diet, you actually have, again, kind of in line with the results of the Segal Labs work, you get people who respond to that and show an increase in butyrate production. You also have people that don't respond. You actually have people that regress. So they actually, their butyrate production decreases. And he was also able to show that if you take these different groups, specifically the non-responders and regressors, if you then subject them to a set of different interventions to try to push them into higher butyrate production using prebiotics like inulin and pectin, or the introduction of Faecalibacterium as a probiotic, you actually have very different results, suggesting that the intervention that is appropriate for an individual may be very different from one individual to another. And here are some of the distributions showing those results, where you can see the distribution of butyrate for the European diet is much lower than it is for the high fiber diet. As you maybe remember, when we were looking at the butyrate levels before, they were around like 5 units, which again, those were log units, so it's probably around 10 or more. So they're in this distribution. And eating fiber is predicted to increase your butyrate production. Here we can see like the non-responders and regressors, and this heat map is showing the context-dependent effect of the interventions. And so yeah, again, I encourage you to read this paper if this topic interests you. And so you can see how these modeling techniques can be leveraged. And with that, I'd like to thank everyone who helped us make this possible, both my fellow instructor, Alyssa, our advisor, Sean, all of our TAs, and all of the folks in the background that allowed this to run fairly seamlessly. So thank you all so much. And now I'm going to pass it off to Sean for some final remarks.
That wraps up day 2 of the course. So the 2024 microbiome course is now complete. Thanks again to the event sponsors, Applied Microbiology International, the Environmental Health and Microbiome Research Center, or the Embrace Center at the University of Washington, and Science in Seattle. Really special thank you to Alex Carr, our intrepid instructor today, who walked us through metabolic modeling at the community scale for communities that reside in the gut, with a particular focus on the gut-brain axis. But then we also saw some other applications of how these models can be deployed and used and validated. Hopefully you found this useful. We will continue to be available on the Slack channel so you can get additional support and help as the next few days go by. So please reach out if you are having questions or issues or trouble. If you want to learn about ISB and all the other cool stuff we do, you can visit us on the web at isbscience.org. So yeah, just a recap of the day. Right, the first day we learned how to take raw data, how to process it, how to do some analyses. Today we learned how to infer something about the functional outputs of these systems in different contexts, right? If you feed someone a Western diet, or if you feed someone a Mediterranean diet, how is their specific microbiome going to respond to that variation? These types of models allow us to play around with that and get some traction on that. So then, now that the course is over, we're moving on to the next phase of the microbiome series, which tomorrow is the symposium. So tomorrow you're going to hear from 6 different speakers from all over the world who work at the interface between the gut and the central nervous system. And you'll hear several examples of how people are leveraging the microbiome to understand this mediation effect of how our microbes affect our brains. We will kick things off at 9 a.m. tomorrow, same as we have for the last couple of days, 9 a.m. Pacific time or Seattle time. Looking forward to seeing you all there. And with that, we'll end a little early. Have a great rest of your day, and we'll see you here tomorrow.